The cristae is the area of the mitochondria where the electron transport chain takes place to produce ATP from the electron carriers NADH and dihydro-FAD, and also water as a byproduct. Following glycolysis in the Krebs cycle, we are left with 10 NADH molecules and 2 dihydro-FAD molecules. The maximum theoretical yield from each NADH molecule is 3 ATP molecules, and each dihydro-FAD molecule will produce 2. Complex 1, NADH dehydrogenase. NAD is oxidized, and the products of this reaction are oxidized NAD, a proton, and 2 electrons. The 2 electrons move from NAD into complex 1 alongside 2 protons, 1 for each electron, which will pass into the outer membrane. When the electrons pass through the proteins, moving from a higher energy state to a lower energy state, they release energy. This energy is used to pump protons across the inner mitochondrial membrane from the matrix to the outer membrane. This pumping of protons into the membrane increases the acidity of the fluid. The first mobile carrier, ubiquinone, accepts the 2 electrons generated from NAD, but also from complex 2 succinate dehydrogenase, which oxidizes dihydro-FAD. They are carried to complex 3 coenzyme Q reductase, which oxidizes ubiquinone. Each electron is then individually passed from complex 3 coenzyme Q reductase to complex 4 cytochrome C oxidase by mobile carrier 2 cytochrome C. As the electrons pass from coenzyme Q reductase to cytochrome C, a proton is pumped through the complex into the outer membrane. Within cytochrome C oxidase, oxygen is reduced to water. Here, 2 electrons, 1 hydrogen ion, and 1 molecule of oxygen react together in 2 cycles to produce 1 H2O molecule. The movement of protons has created a chemiosmotic potential which can be used to synthesize ATP.